everyone, and thank you for joining us today. I'm Melinda Zeno, AGRF's Genotyping Manager, and welcome today to AGRF's third webinar on our series for complex diseases. Interrogation of complex diseases, as most know, can be multifaceted, and this webinar series focuses on using the AGRF's array genotyping service for such complex cases. And what can be more complex than an individual's health and fitness? So array genotyping provides scalable and cost-effective solutions, and one such array is the Illumina Global Screening Array, which features over 650,000 markers in a 24-sample format. Today, we're extremely excited to welcome today's speaker, Dr. Cristina Fortuno, who will be presenting her talk on genetic testing and health and wellness, which is a commercial experience using the GSA. Cristina holds a Master in Science and Biotechnology from the Polytechnic University in Valencia, Spain. And after gaining experience working in reproductive genetics, she decided to move to sunny Brisbane to complete her PhD in human genetics at the University of Queensland. Cristina is currently working as the product development lead at Fitgenes, which, for those who don't know, is an Australian-based genetic testing company focused on health and wellness. This role combines Cristina's interest in the use of genomics in healthcare as a tool to improve diagnosis and deliver personalised interventions, but also discovering more about a person's predisposition to certain health traits. I'm incredibly excited to hear Cristina explain how these variants in your genes can influence your health and fitness, and hopefully everyone else today is as well. After today's talk, I'll invite attendees for questions. We can do this via the chat function, or you can use the raise your hand function as well. So please join me today in welcoming Cristina for her presentation, and thank you. Over to you, Cristina.
Thank you. I will share my screen now. Give me a second. Thank you very much for having me in this complex diseases webinar series. As Melinda mentioned, I will be talking about genetic testing for health and wellness, and in particular, our experience using the global screening array. During this presentation, I will give you a bit of background. I will talk about Fitgenes and some of our products, and then I will move to the global screening array and some conclusions. I will start with a brief background so you understand more what's the motivation behind Fitgenes. Nowadays, we live longer, but that doesn't mean that we live healthier. And in the current society, there is a high prevalence of obesity, which is a risk factor for many diseases. There is also a high prevalence of many chronic preventable diseases, for example, diabetes, cardiovascular issues, metabolic disorders, and all of this has an increasing cost burden on society. All of these diseases or health issues always have a genetic component, and therefore genetics can be a tool for risk prediction and prevention. With this in mind, Fitgenes was established in 2009, and it offers a range of DNA tests that are available mostly through accredited health practitioners. It's focused on predisposition, so we don't do diagnosis, we focus on predisposition to obesity, diet-related health issues, and cardiometabolic health. It operates mostly in Australia, but also in New Zealand and some parts of Asia, and increasingly in Europe and the US. And we also make use of a proprietary technology platform that the practitioners can use that is TGA listed, and this is useful to order the DNA kits, to review the results, or to obtain additional information about each of the genes or interventions or all the references. Genetic profiling has many benefits because if you can find out your risk for a health issue, then you can do something about it. You might prevent it. You can also get personalized and therefore more efficient treatment. It's also useful to differentiate what's genetic or what's cultural, social, or environmental, although in many cases it's a combination of both. And also genetic testing is getting more and more common, so you can reach more people with this tool. In our products, we have some basic criteria to choose genetic variants to be included in the report. We usually prefer genes that have a known function. We also usually prefer genetic variants that are relatively common in the population. The associations of the genetic variants with any trait should be backed up with enough evidence-based research. And finally, the results should be actionable. Now I will describe some of our products so you can get a better idea about the type of genetic testing that we do. I will start with health and well-being. This is a genetic profile of 64 genes that are involved in different physiological functions relevant to human health that I will explain later. And this is mostly focused on genetic predisposition to a range of multifactorial health issues that by definition are affected by the environment, and therefore you have the opportunity to recommend personalized lifestyle interventions. This one is only available through accredited health practitioners. The sections of this report are the following. There is a section of lipid metabolism and cholesterol regulation. So we test genetic variants in a number of genes included in this process. For example, we test a gene that encodes the beta-2 adrenergic receptor that binds adrenaline, and a genetic variant has been shown to cause slow receptor status, and this is associated with reduced fat burning during exercise, accumulation of body fat, and therefore higher BMI. However, other studies in the literature have shown that people that have this genetic variant can benefit from high intensity exercise because this can activate the receptor. There is also a section on inflammation in which we test genes that encode pro-inflammatory and anti-inflammatory cytokines. Another section on cellular defense in which we test mostly genes that encode phase 1 and 2 enzymes that are involved in the detoxification processes and drug metabolism. There is also vitamin D metabolism. Another section on cardiovascular biology in which we test genes that are part of the renin-angiotensin system, which is involved in the regulation of blood pressure. And some genetic variants in these genes have been associated with an increased enzyme activity and therefore a higher risk of high blood pressure. And finally, there is also a section about genes that are involved in methylation and homocysteine metabolism. Another product that we have, it's called Food Choice, and this is focused on predisposition to dietary intolerances and sensitivities. This one is also only available through accredited health practitioners, and it has 5 different sections that I will explain now. One of the sections is about celiac disease risk, because it's known that there is a link between the human leukocyte antigen region, which is involved in the regulation of the immune system, and celiac disease. And a number of specific genetic variants have been identified and validated to be associated with a slightly increased risk of celiac disease. Therefore, this test, if you get like a positive genetic result, is not a diagnosis. This indicates a slightly increased risk in comparison to the rest of the population. But here, a negative result is very informative because this can almost rule out the risk of having celiac disease. That is very useful in cases when diagnosis with other methods is not clear or when there is some family history. Other 2 sections of this food choice report are about lactose and alcohol intolerance risk. And here we look at genes that are involved in the metabolism of lactose, in particular the LCT gene that encodes the enzyme lactase, which is involved in the digestion of lactose, and also other 2 genes that are involved in the alcohol metabolism process. And in particular, we test specific genetic variants that have been associated with lower gene function, and they are associated with predisposition to lactose and alcohol intolerance. And finally, the other 2 sections of this report are about caffeine and salt sensitivity risk. And here we look at genes that are involved in the caffeine metabolism pathway, and also the genes that are part of the renin-angiotensin system that I mentioned before that is involved in the regulation of blood pressure. And we test specific genetic variants that have been associated with different health issues following caffeine and salt intake. At the end of this food choice report, we provide a summary table that looks like this, in which we show the categories of the report, the genetic variant tested, the result, the meaning of this result, and then some interventions. For example, for this person, they have a genetic variant that is associated with increased risk of caffeine sensitivity, and therefore we recommend to decrease caffeine intake or to limit the intake to the morning or early afternoon. I would also like to mention a product that we have that it's called Carb Choice, which is focused on predisposition to obesity. Here we look at the AMY1 gene that encodes the salivary amylase enzyme that is involved in the digestion of starchy carbohydrates. And multiple studies in the literature have shown a link between the AMY1 copy number and predisposition to obesity and type 2 diabetes. In particular, people that have a lower copy number have lower gene expression and more difficulty digesting this type of carbohydrates. This report also gives personalized advice on diet and exercise. And this one is available through health practitioners, but you can also get it direct to consumer. Then we have a vitamin D report that, as the name indicates, it's specialized in vitamin D metabolism. And we look at genes that are involved in different vitamin D process. For example, genes that encode vitamin D receptors, genes that encode enzymes involved in converting vitamin D to its active form, and proteins involved in transporting vitamin D. And in particular, we test genetic variants that have been associated with different vitamin D levels. And similarly as before, this report also provides additional information about, in this case, vitamin D and health, including vitamin D deficiency, other factors affecting vitamin D levels like sun exposure or diet, and we also provide some sources of vitamin D. And this one is again only available through accredited health practitioners. I would also like to mention other products that we have in development, one that might be called Diet Choice, and this one will be focused on eating behavior and obesity predisposition, and will have different sections. One will be about genes that are involved in satiety and hunger level. For example, the receptor for leptin, which is the satiety hormone, then the MC4R gene, which is the most common cause of monogenic obesity, and also the FTO gene, which is the fat mass and obesity associated gene and has been termed the fat gene. And then we will also look at genes that are responsible for the perception of different tastes, and all of this can affect the eating behavior of a person and potentially some type of predisposition to obesity. We are also working on a report completely focused on immunity, and we are looking at the literature to select genetic variants that are associated with predisposition to different infectious diseases or severity of these diseases. And I would also like to mention, although we don't use GSA for this, but if a patient already has data from 23andMe or Ancestry, we can convert it to most of Fitgenes products, in particular to all products except Carb Choice, which is copy number. Now that you are more familiar with our products, how do we do all of this? How do we do the testing? Our partner lab is AGRF, that you probably know, and they offer a range of service, including DNA sequencing using the Illumina Global Screening Array, which is what we use for testing. I have to apologize, but my cat is on the way. The main features of this array are the following: is a next generation genotyping array. It incorporates more than 600,000 markers, and you can also get additional custom content. It's very widely used for a range of clinical research applications. This is risk profiling studies, pharmacogenomics research, wellness characterization, and complex disease discovery. It allows to use 24 samples per array, and the samples can be from different origins, for example, blood or saliva. GSA has actually been quoted a lot in the literature. For example, this study, which is the first proof of principle study to use GSA to detect rare variants. And here they aim to test the potential of a customized GSA. So here they add an additional 9,000 variants to diagnose primary immunodeficiency disorders for only €40 per sample. And they achieve an accuracy rate very close to 100%. Sensitivity to detect rare disvariants is also high, 87%, and the diagnostic yield is 39%. And they say this can be easily increased by adding more variants to the custom content. And then they conclude that this customized GSA can be a promising first-line rapid screening tool for primary immunodeficiency disorders. This other study starts talking how in the past maybe GWAS didn't achieve their full potential because they didn't select the right genotyping array. So here they do a comparison of the coverage between 3 different Illumina arrays in relation to inflammatory and immune-related pathways. And they find that the GSA performed the best in comparison to the other 2 arrays in relation to global coverage and also coverage for most chromosomes, for most considered pathways, and for most genes. Other studies don't look specifically at how well GSA works or performs, but they mention as part of the methods. For example, this one, in which they aim to correlate variation in genes involved in drug metabolism with disease outcomes, using data from 250 breast cancer patients, and they find 2 significant associations that could be used as biomarkers for a bad outcome in breast cancer patients. After all of this, what's the benefits of this global screening array for us, for Fitgenes? As I mentioned before, it includes a large number of expertly selected clinical research genetic variants. So this allows us to test for all the variants that were already included in our products before using GSA, and also gives potential to create many new products that are in line with Fitgenes' goals. It has been shown to have a very high sequencing accuracy rate. It allows labs to cost effectively process hundreds of thousands of samples a month. In relation to our previous provider, this has a better pricing on contract and also better pricing flexibility. And we have also seen that AGRF can cope very well with low DNA quantity thanks to their concentration protocols, and they are not accredited, which is of course very good. In conclusion, there are a large number of genetic association studies in the literature, and DNA sequencing technologies keep evolving, allowing higher accuracy and lower cost. And at the same time, people are more and more interested in genetics. Therefore, this gives the potential in using genetic testing to find out an individual's predisposition to different health issues or traits, and therefore the opportunity to recommend personalized interventions and achieve health outcomes. It's of course essential to be critical with the selected studies, and we always select genetic variants whose associations have been validated by multiple studies. And in conclusion, the global screening array is a reliable and cost-effective sequencing platform that has multiple applications and that totally meets Fitgenes' needs at this time. Thank you very much for your attention. And also I put my email in case someone has some question later or wants to collaborate somehow.
Thank you very much, Cristina, for a very interesting talk this afternoon. So I'll put it over to our attendees today. If anyone has any questions, as I said, you can use the chat function or you can use the raise your hand function in the chat function.
Cristina, I'd just like to ask a quick question. That was a wonderful talk. Just wanted to know what your thoughts are on where you think personalised genomic testing is going, what the future holds for it.
Well, I think it's getting more and more common, and definitely I think this is going to be a very popularly used tool in the health space. So it has multiple applications, and I think it will be more and more used in many applications, but mostly the healthcare space.
Thank you.
Cristina, it's Melinda here. So are you finding at Fitgenes that the perception in the public is becoming more acceptance to personalised genomics, and that there's not the apprehension that there was a few years ago, and that you're seeing more people decide to partake and find out more about their own health and fitness?
Yeah, I can see that people believe more and more in genetics. Of course, you have to always show that the references and the evidence base, but definitely people are more open to genetics.
Well, if there's no more questions for today, thank you very much, Cristina, for a very interesting talk this afternoon. It's given me a lot more insight into my own health and fitness, especially with the Christmas period coming up. I think we all should be more mindful of everything. Maybe I do need my genetics to let me in, my traits to do the talking, whether I should go for that extra coffee in the morning or that extra in the evening. So thank you very much. And if there's no more questions, I'll wrap it up here and we'll let Cristina get on with her day. So thank you very much, everyone, for attending today. And join me in thanking Cristina for a wonderful presentation.
Okay, thanks again. Bye.
Thank you.
Thanks, Cristina. Bye-bye.